So I'm just going to go ahead and get us started while people are kind of still trickling in. So thank you so much, like I said, for being here tonight. For those of you who don't know me, my name is Kate, and I'm the VP of Research, Impact, and Engagement here at CTF, and your monthly host for the NF Knowledge series. We host these almost every month around different educational topics that we think are important to the community, and we're happy that you're here with us. We do have closed captions available as always by a live captioner. You can make sure those are turned on by clicking that CC button at the bottom of your Zoom screen. And so you can get those turned on. We are recording this event. We always get that question, and I want to make sure I say it several times. This is being recorded, and it will be made available on our CTF YouTube channel. There may be some redacted images of slides because there is data that Dr. Kim will be sharing that can't go up on a YouTube site, but the presentation itself will be available to view later at the Children's Tumors Foundation's YouTube site. And just so you know, I'll be making announcements at the end of the webinar today that include links to different things, but don't worry, we'll be pasting everything into the chat multiple times to make sure that it's clear and you can grab those links or copy and paste things and keep that information for yourself. So I'm very pleased now to introduce, we have a special guest with us tonight. John Morris is a member of the Children's Tumor Foundation Board of Directors and an NF2 dad. John's advocacy in this space is impressive, to put it mildly, but I'm thrilled to have him here with us tonight. So John, I will hand things over to you.
Thank you, Kate. I appreciate the introduction, and thank you to all of you for joining. My name is John Morris, and I'm a board member at the Children's Tumor Foundation. I apologize for the lighting in my hotel room. I think I look a little washed out, but nonetheless, I'll press on. I'm really grateful to be here with all of you in the NF2 community. This is a really personal endeavor for me. I'm an NF2 dad. My son lives with NF2 related schwannomatosis. And I'm especially thankful that we're here to talk about progress that's being made. I've been associated involved with CTF for well over 10 years now, and it's been just fascinating to watch the science grow and for us to get to a place where we can start talking about making meaningful progress in ending NF. So you may know that CTF is proud to be an early investor in one of the programs that you're going to hear about tonight. This is exactly the kind of targeted investment that we're trying to make, which is patient-focused work, and that's what a CTF exists to do. We are all in on NF2. We find, drive, invest, and advance the strongest drug programs that we can, and we push them into clinical trials so they can reach patients much faster than they would through traditional means. Thank you to the patients and families who make all of this possible, and to the researchers, clinicians, and PRG Science and Technology for advancing this important work. And thank you to all of you and everyone who's here tonight. NF2 needs more treatments, and we're here to deliver them together. Now, it's my pleasure to introduce our guest. Our guest tonight is Dr. Minju Kim. Dr. Kim is the Director of Clinical Development at the PRG S&T. She's been with the PRG for 9 years, and she holds a PhD in microbiology from Pusan National University in Korea. She's going to talk to you about some of her recent work. Dr. Kim, thank you for being here tonight. I'll turn it over to you.
Thank you for your introduction, John Morris. And I would like to share my slide first. Can you see my screen, everyone?
Yeah, looks great.
Okay, thank you. Hello, everyone. My name is Minju Kim, and I lead the Clinical Development Center and PRG S&T. First, I would like to sincerely thank CTF for giving us this opportunity to introduce Trineumin, our new investigational drug for NF2. Today, I will share the story behind the Trineumin, how we designed our clinical trial in Korea, and some preliminary data on how it's working for our NF2 patients. I'm sorry. Let me briefly introduce our company. PRG-SNT was founded in 2017 in Busan, Korea. We are a team dedicated to find treatment for rare disease that currently have no cure. Our founder and CEO is Dr. Bum-Jun Park. He is a professor of molecular biology at Busan National University. He is a leading expert who has published numerous papers on rare disease. Throughout his career, he has successfully completed 23 technology transfers and holds more than 20 patents in this field. Currently, PRG-SNT consists of 36 dedicated employees. Our organization is structured into three main divisions: the R&D, clinical development, and management division. This specialized structure allows us to efficiently manage everything from early drug discovery to global clinical trials. This is an overview of our R&D pipeline. Currently, we are developing treatments for several indications using 3 core compounds. The second one on the list is Trineumin, our NF2 treatment. I'm very pleased to share that we are now on the verge of completing phase 1 clinical trial with the NF2 patients in Korea. Now, let's move on to today's main topic, NF2. I believe most of you joining us today are already very familiar with this condition. As you know, NF2 is caused by an NF2 mutation in the NF2 gene. This leads to the growth of benign tumors such as meningiomas, vestibular schwannomas, and ependymomas throughout the nervous system. These tumors can press on nerves, which often result in hearing loss, vision problems, and other neurological challenges. Although it is a rare disease affecting about 1 in 33,000 people, but to the person living with it, it is 100% of their reality. The impact on the daily lives of patients and their family is very significant. To help you understand how Trineumin works, I would like to explain its mechanism of action. In a healthy state, the NF2 protein forms a complex with another protein called RKIP. This complex acts as a natural brake to stop the abnormal growth of schwannoma cells. Also, the two receptors, TGF-beta receptor 1 and 2, stay in a perfect balance, allowing normal cell signaling to function correctly. However, in the NF2 deletion condition caused by the NF2 gene mutation, this balance between the two receptors is broken. This imbalance causes RKIP to bind abnormally to the TGF-beta receptor 1, which unfortunately leads to the degradation or destruction of RKIP. Since the loss of RKIP is what leads to tumor formation, our team searched for a solution. We wanted to find an inhibitor that specifically blocks this destruction of RKIP without disrupting other healthy cell pathway, and that is Trineumin. When you treat a cell with Trineumin, it selectively binds only to TGF-beta receptor 1. This prevents RKIP from the binding to it, effectively blocking the degradation process. As a result, the balance between the receptor is restored, RKIP is saved, and the tumor growth is inhibited. This eventually encourages Schwann cells to develop normally again. Now let's look at the data from our animal model studies, where we tested the preventive effects of Trineumin. We administered Trineumin orally to NF2 model mice from the age of 11 weeks up to 13 months. The results were very encouraging. we observed that the formation of tumors in the DRG was significantly inhibited. For those who might not be familiar, the DRG dorsal root ganglion is a cluster of nerve located near the spinal cord, and it is common site where tumors tend to develop in NF2 mice. Furthermore, we confirmed that Trineumin not only saved RKIP, but also increased the expression of SOX10 and MBP. These are key markers that shows Schwann cells are maturing normally. This suggests that Trineumin doesn't just stop tumors, it helps the nervous system stay healthy. This slide shows the result of whole body micropacity analysis after administering Trineumin to NF2 model mice. Please take a look at the images. The red arrows here points to where the tumor has developed, and while the white arrows represent the background signal. When you compare the control group, which received no Trineumin, to the Trineumin groups here, the difference is clear. The red shaded area indicated by the red arrow represent the tumors. As you can see in the Trineumin treated group, this red area is mostly cleared. In the mice treated with Trineumin, tumor formation was significantly inhibited throughout the head and the entire body. We also observed a dose-dependent effect, meaning the result got even better as the dose increased. Most notably, in the head area, tumor was almost non-existent at dose of 50 milligrams per kilogram. Through this result, we were able to clearly confirm the preventive effects on Trineumin. This visual evidence gives us strong confidence in the drug's potential. Next, we observed the therapeutic effects in our NF2 model mice. For this study, we administered Trineumin via IP injection twice a week from the 9 months to the 13 months. As a result, we found that tumor formation in the dorsal root ganglion was effectively inhibited compared to the control group. Most importantly, we were able to confirm that expression of RKIP as well as Schwann cell differentiation markers, SOX10 and MBP, significantly increased. This confirms that Trineumin is effective not only in prevention, but also in treating existing conditions by helping cells develop normally. For those interested in more in-depth information, you can find our full dataset published in the Neuro-Oncology Journal. Now, I would like to share the result of the various non-clinical studies we have conducted so far. To ensure the safety of Trineumin, we performed 4 key safety pharmacology studies. We confirmed that the drug is very safe, with no significant impact on the respiratory, cardiovascular, or central nervous system. In addition, Trineumin showed excellent metabolic stability. It also demonstrated moderate protein binding and good permeability, which are important factors for how the drug travels through and is processed by the body. This result provides a very solid foundation for its safety profile. Moving on, we confirmed that Trineumin has almost no inhibitory effect on CYP enzyme, which are the primary metabolic enzymes in the liver. We also found no evidence of genotoxicity. Regarding our tox studies, we have already completed the 4 weeks and 13 weeks repeated dose tox studies. We are currently conducting long-term tox studies, 26 weeks in rodents and 39 weeks in the non-rodents, which we plan to complete within this year. Most importantly, for the families watching today, we are currently conducting juvenile tox study to prepare for potential use in children. This is expected to be completed in the first half year of this year. We also plan to start a carcinogenicity study later this year. Based on all the non-clinical data collected so far, we believe Trineumin is a very safe and reliable drug candidate. As I mentioned earlier, since Trineumin binds to the TGF-beta receptor, we wanted to make sure it doesn't accidentally interfere with other important proteins. To confirm this, we conducted a kinase panel assay, testing it against 435 different kinases to check for any off-target effects. The result showed no inhibition of these kinases, and even the TGF-beta1 receptor was barely affected. This means Trineumin is very specific and doesn't disrupt other vital cell signals, unlike other kinase inhibitor. Also, because NF2 tumor grows in the nervous system, it is crucial for the drug to reach the brain or spinal fluid. We performed blood-brain barrier penetration test and confirmed Trineumin crosses the barrier effectively. We detected the drug in both the brain and the spinal fluid, proving that it reaches the exact locations where it's needed most. Now, I would like to move on to the most important part, the clinical trial we have been conducting in Korea since June 2024. This is where we see that Trineumin may actually be helping near NF2 patients. Currently, we are conducting a phase 1 part 2A clinical trial in Korea titled an open-label dose-finding study to evaluate the efficacy and safety of PRGN-01 in patients with NF2. The study is being held at Asan Medical Center with Professor Bom-hee Lee as a principal investigator. I'm happy to announce that the first cohort, the final dose group of phase 1, is expected to complete their final administration by the end of this January. This phase 1 part of the study involves a total of 16 patients. Our primary goal is to evaluate the safety and tolerability of Trineumin and to determine the MTD, maximum tolerated dose, and RP2D, recommended phase 2 dose. Additionally, we are exploring the drug's pharmacokinetic properties and its initial efficacy. In simple terms, this means we are studying how the drug travels through the body, how well it is observed, how long it stays in the system, in the body to fight the tumors, and how it is eventually cleared. Understanding this helps us find the most effective way to dose the medication for each NF2 patient. Once the RP2D is determined, we will immediately begin phase 2 trial, which will involve a larger group of NF2 patients. This next phase will focus on clearly confirming the therapeutic efficacy of Trineumin. We are moving forward as quickly, as safely as possible. This diagram shows our clinical trial design. As you can see, the protocol originally included 6 dose groups. However, the third and fifth groups were set up as buffer groups in case we needed to reduce the dose due to safety concerns. I'm very pleased to report that we have seen no significant adverse events so far. Because of this excellent safety profile, we were able to proceed directly through the 4-dose group highlighted in yellow. Currently, we are administering the final 150 milligram dose to cohort 6. In this study, we perform a DLT, dose-limiting toxicity assessment, after 12 weeks of treatment to ensure patient safety. We expect to complete the DLT evaluation for this final group by early February. Once this is complete, phase 1 will officially conclude. We will then use all the data collected to determine the RP2D and begin moving into phase 2 immediately. In this clinical trial, we are measuring the efficacy of Trineumin through 3 main items. First is the radiographic response. We use MRI scans to track changes in tumor size and see how effectively Trineumin is working. Second, we are monitoring functional improvements in hearing. We performed WRS and PTA tests to see if our patients' hearing is being preserved or improved. Third, we are evaluating the quality of life throughout 3 different patient surveys, because how our patients feel in their daily lives is just as important as the clinical data. Today, I would like to briefly share some preliminary data from our ongoing trial focusing specifically on the change in tumor size seen through MRI scan. This slide presents MRI data from our first cohort. First, let's look at the graph here. The horizontal axis shows the treatment period in months, with 0 representing the baseline. The point just before Trineumin was first administered. The vertical axis indicates the percentage change in tumor volume. Our study is designed to allow for dose escalation. Even if a patient starts at a low dose, they can move to higher dose once safety is confirmed in subsequent cohorts. For instance, the patient in cohort 1 started at 25 mg. As the trial progressed and safety was confirmed for higher doses, this patient was escalated to the 50 mg and then to 100 mg, which is their current dose. Due to the accelerated titration design of the first cohort, only 1 patient was assigned to this group. This patient had 3 different types of tumors. First, looking at the vestibular schwannoma, at the initial 25 mg dose, it shows a tendency to increase here. However, after the dose was increased to 50 mg, we began to see a downward trend. Moving on to the non-vestibular schwannoma, we haven't seen a significant response yet, but most notably, in the case of meningioma, we saw a very encouraging result here. Within just the first 3 months treatment at 25 mg, the tumor volume decreased by nearly 20%. Since then, we have observed a sustained reduction of over 28%. Unfortunately, due to hearing loss caused by vestibular schwannoma, this patient underwent cochlear implant surgery last October. Because of the resulting imaging interference from the implant, we can no longer measure the meningioma volume through MRI. However, the data gathered up to the point clearly demonstrates the drug potential efficacy. Now let's look at the data from our second cohort, which is started at 50 milligram dose. This group consists of 3 patients, 01, 02, 04, 05. Similar to the first cohort, these patients also began their treatment at 50 milligram, but once the safety of 100 milligram dose was confirmed, their dosage was escalated. Currently, all 3 patients are receiving 100 milligram of Trineumin. Let's look at the individual results. First, for patient number 02, who has 2 types of tumors, both of vestibular schwannoma and non-vestibular schwannoma, shows slight fluctuations, but they are maintaining stable status within a 20% range. Next, patient 04 has only non-vestibular schwannoma. So far, we haven't seen a significant response to the drug yet, and the tumor are showing a tendency to increase. Finally, for patient 05, the meningioma is showing a downward or regressive trend here, staying with the 20% range. Meanwhile, the non-vestibular schwannoma tumor in this patient are remaining stable. This early observation helps us understand how different tumor types respond to increasing doses of Trineumin. Next is our first cohort, the 100 milligram dose group. This group consists of 6 patients who have been receiving treatment for an average of about 8 months now. Overall, we are seeing the patients are maintaining stable conditions. Furthermore, in several specific tumors across different patients, we have observed a clear downward trend or reduction. Finally, I would like to share an update on cohort 6, our 150 milligram dose group, but there is no graph because we didn't conduct the MRI scan yet for cohort 6, 150 milligram dose group. But administration for this group began last October. We expect to obtain the first 3 months MRI result sometime in late January to February. Given the dose-dependent responses we've seen so far, we are very hopeful and optimistic about seeing even stronger efficacy data from this higher dose group. Regarding long-term safety, some of our patients have now been taking Trineumin for over 18 months. During this period, we have closely monitored all side effects. So far, we have recorded 10 adverse events across 7 patients. However, only one of these fatigue was found to be related to our drug Trineumin, but most importantly, this was a grade 1 event, which is the mildest level and not a serious concern. Excluding that case, the remaining events showed no or low relevance, with some being not accessible. Furthermore, all of them are grade 1, and we were observed no serious adverse event to date. Based on this data, we are confident that Trineumin is relatively safe and well-tolerated drug even for long-term use. To summarize everything we've discussed today, first, Trineumin is a first-in-class drug and selectively binds to TGF-beta1 receptor and restored ARKIP levels, effectively regressing tumors in NF2-related schwannomatosis. Second, our non-clinical study have shown a very favorable safety profile with no genotoxicity and no significant interaction with other drugs. Third, in our ongoing phase 1.2a clinical trial in Korea, we have secured a favorable safety profile for those up to 100 milligram. We are currently in the middle of the 3 months administration of the 150 milligram dose. We expect to collect the safety and efficacy data for this final group by late January and review the final result in February. Finally, after reviewing the 150 milligram result in February, we will hold an SRC Safety Review Committee meeting. There, we will decide whether to conclude phase 1 and determine the RP2D to start phase 2 or to proceed with further dose escalation to ensure the best possible efficacy for our patient. We are committed to moving forward with utmost care and speed for the NF2 community. This diagram illustrates our projected clinical development timeline. As I mentioned, if we decided to conclude phase 1 trial after reviewing the 150 milligram results, we plan to transition immediately into phase 2 at Asan Medical Center in Korea, starting around the second quarter of this year. In addition to our progress in Korea, we are actively preparing for global clinical expansion. In the United States, while not finalized, we are in discussion with MGH and to another site to initiate investigator-initiated trial. Furthermore, we are continuously consulting with institutions and investigators to expand the clinical trials into the UK, Europe, and China. Our goal is to make Trineumin available to the global NF2 community as swiftly as possible. That concludes my presentation for today. I would like to express my deepest gratitude to the CTF for providing me with the opportunity to speak today and for their generous funding of our clinical MRI imaging. I also want to extend my sincere thanks to the KDDF for our essential financial support for the entire clinical trial process. With your continued support, we are able to move forward on this journey toward a cure. If you have any questions, please feel free to contact us through our company website or via email. We will make sure to review each inquiry and get back to you with a detailed response. Thank you once again for your time and attention today.
Thank you so much, Dr. Kim, for being here with us and for taking the time to walk everyone through your clinical trial and where things stand currently. I just wanted to take a moment to let our attendees know that out of respect for the fact that the language, Dr. Kim's primary language not being English, that we decided not to do a live Q&A just so that any questions could be carefully considered and responded to accurately. So that's why she's generously shared the email address if you want to reach out to them directly. My email will also be in the chat shortly, and you're welcome to reach out to me. And any questions that were shared tonight in our chat or in the Q&A have been recorded so that we can get back to you with answers to those as well. I also want to just take a moment to say that there's a lot of talk. Obviously, we were talking about a clinical trial. And there's a lot of language around clinical trials that is not necessarily familiar to everybody. So I'm also going to share a link in our chat that takes you to just a pretty simple explanation of clinical trials that we've developed that lives on our website in our resource library. But I thought it might be of particular interest tonight for those of you who are curious about a phase 1 clinical trial versus a phase 2 and what does that really mean. So that will be there for you as well. A big thank you for joining us to everyone who made the time to be here tonight. We're also sharing some other links in the chat, specifically directly to our page that's dedicated to the progress we're making in finding treatments for NF2. So that will be in the chat. I encourage you to take a look if you haven't checked that out in a while. We keep that updated with news. Again, that clinical trial info sheet is in there. I want to remind you that we have the Knowledge Series monthly, and to check our website, we have a whole calendar where you can see what's coming up. It'll be updated very soon, so you can see kind of the first 6 months of the year, what we'll be talking about and when. And just a quick shout out to say that we will be having a pretty special NF Knowledge Series webinar on February 28th in honor of Rare Disease Day. So be paying attention and looking for details about that coming very soon. I hope to be able to introduce everyone to the relaunched NF registry at that time. So there'll be more details coming soon. To make sure you get those details, be sure to sign up for the CTF newsletter. That is the best way to know when things are happening that you could be a part of and to kind of hear all of the updates in research and clinical care. And like I said, if you have questions, which I'm sure you do about tonight's presentation, please don't hesitate to reach out to me directly or to the email that Dr. Kim provided or both. We're happy to talk with you and make sure that you understand and have the answers that you are looking for. So a big thank you again for everyone for being here. Thank you to Dr. Kim for your lovely presentation. And thank you to John Morris for being here to welcome everyone and share a little about your involvement. Really appreciate that. And we hope to see everyone again in February. So have a great night. Signing off. Good night, everyone.